I was just telling Bill Green, whom you're going to meet very shortly, that identifying some of the speakers were derived from, I was helping teach a MD PhD course, and we were reading some of these papers from our experts today, and I got excited about inviting them. That's one way to bring together some exciting developments in research. You read the literature and find out who to bug. Daniel Salzman is in the Department of Psychiatry and Neuroscience at Columbia University, Associate Professor of Psychiatry and Neuroscience in the Kavli Institute for Brain Science at Columbia. He received his doctoral degrees at Stanford University. Research is focused on the neural basis of cognitive and emotional behavior. You see the title on the slide. Welcome, Daniel. Thank you.
So good morning, everyone. It's a pleasure to be here, and it's always fun to be able to talk about one's work and one's passions with audiences that it really will impact the future of the field in a very different way than the usual audience that I speak to, because whether you're a teacher or just a young student, you have the opportunity to help encourage people to pursue careers in neuroscience research. And as a psychiatrist, I believe that kind of long-term investment is what's really required if we're going to ultimately make a difference and be able to treat mental disorders in very effective ways. So it's my pleasure to tell you about some of my work and also some of the work that goes on in the field today, trying to understand basic mechanisms of the brain that help mediate emotion and emotional behavior. And obviously I'm interested in this because I am a psychiatrist as well as a neuroscientist, but there's another secret to my background that helps explain my somewhat passion and interest in this general topic. And the general topic concerns how do stimuli in the world all of a sudden acquire emotional meaning, and how do I learn to respond to those emotionally meaningful stimuli in an appropriate way. And so this is the example from the other aspect of my life that helps explain my interest in pursuing this area of research. So the question is, how do you feel when you view this kind of stimulus? And this, for some people, may not elicit a strong emotional response, but for me, and because I'm a tremendous wine enthusiast, it elicits a very strong positive emotional response. This is a bottle of red Burgundy from a producer named Armand Rousseau. It's one of the most beautiful labels from Burgundy in France, and it's from a vineyard called the Chambertin, which is one of the most famous vineyards in Burgundy. And if you're lucky enough to have drunk this wine many times with friends and family, and in my case, I've also visited the domain a number of times and had the wine with the wine producers, just seeing this image of this bottle brings all kinds of positive emotions to my way of viewing the world. And but this is just an arbitrary visual stimulus that until I had those experiences, it was devoid of that kind of emotional meaning to me. This is another arbitrary visual stimulus. This is a bottle of wine that a friend gave me for a gift. They did not know me very well. I wasn't even willing to cook with this. So it's another arbitrary visual stimulus that now has a very different meaning. It's acquired through other experiences telling me that I really don't like what comes out of that bottle. So the question then before us in some ways is, how does the brain acquire and assign these emotional meanings to these kinds of stimuli? And that's the kind of problem we're interested in, not just because of my passion in wine, but because we believe that understanding how the brain implements these kinds of processes will have a very large impact on trying to understand how psychiatric disorders arise. And the notion would be something like the following. If your car is broken, you would never want to take it to an auto mechanic who doesn't know how the car normally operates. You got to know how the car normally operates before you can start to really diagnose what's wrong with the brain. And so there's a lot of investigation in neuroscience out there that is not going to pay dividends tomorrow. They're going to pay dividends over a longer time scale in trying to understand how the brain operates so that then we can understand better how it becomes dysfunctional. And the basic process that I'm going to be telling you about, which is how sensory stimuli acquire meaning, emotional meaning, is a process that clearly goes awry in many different psychiatric disorders. So for example, when you think about schizophrenia, many people think of patients with schizophrenia and instantly think of the delusions and hallucinations that often characterize the disorder for them, and those are referred to as positive symptoms in schizophrenia. But there's another whole spectrum of symptoms in patients with schizophrenia called negative symptoms, And these really have to do with, in some ways, an emotional disconnect that these patients have with the environment around them. So they lack motivation. Sensory stimuli that would normally produce a certain type of emotional response fail to do so. And so there's a dysfunction in the neural circuits in the brain that are linking sensory stimuli to motivational significance in patients with schizophrenia. Now, this is obviously also true in depression. I mean, there's a saying, people that are depressed always see the glass as being half empty rather than half full, right? So this has to do with the manner in which you assign emotional meaning to stimuli. And so patients with depression take stimuli and process stimuli that normally would produce pleasurable experiences, but then fail to produce those pleasurable experiences when they encounter those stimuli. And again, this is probably due to dysfunction in the neural circuits that are linking our sensory world, our perceptions of sensory stimuli, with our emotional world, the meanings that they derive. And obviously, another big category of psychiatric disorders where dysfunction in this process plays a role are anxiety disorders. And in anxiety disorders, like post-traumatic stress disorder or PTSD, sensory stimuli acquire the ability to generate enormously stressful and anxious experiences for patients. And in post-traumatic stress disorder, that might be experiencing gruesome scenes, gruesome activities during war, and then even when the veteran comes back to home and encounters stimuli that are somewhat similar to those stimuli they experienced at war, instead of being able to regulate their emotional responses to those stimuli, they fail to regulate those emotional responses to the stimuli and get a whole spectrum of anxiety and other symptoms in response to them. So it's dysfunction in these neural circuits that link emotional meanings to sensory stimuli that really go awry in virtually every single neuropsychiatric disorder that you can imagine. So the question is, how are we going to become better automechanics? How are we going to get to the point where we can understand how the brain operates better and use that to gain insight into how we can develop treatments for psychiatric disorders? And in many ways, many of the biomedical investigations into this problem really originate in some ways from work that was done by Darwin many years ago. And Darwin wrote a book in 1872 called The Expression of Emotions in Man and Animals. And here's some figures from that book, and you can note the similarity in facial expressions here between man and animals. And so one of the main insights to come from this book is that the ability to study emotion and understand emotion in humans can be greatly assisted if we use animals as model subjects for trying to understand how emotion operates in the animals. And the insights that we gain from studying animal models can then be used to understand how the human brain is functioning. And so this insight really forms the basis for much of the biomedical research. In the first talk today, we heard a lot about using mice during the study of different processes involving emotion and cognition. Now, one of the other main insights that's particularly relevant for emotion that came from Darwin's book was the fact that part of what an emotion is the bodily expression of emotion, and that one key component of that bodily expression of emotion is what are so-called autonomic responses that are an intrinsic part of the emotional experience. So what are autonomic responses? Autonomic responses are things like when your heart starts beating really fast, when your palms get sweaty. These are controlled by a part of your nervous system that's called the autonomic nervous system, and they are adaptive responses that you use in order to try to maintain some sense of homeostasis in a threatening or changing environment. And so, and if you think about it, when you're really scared, you get autonomic responses. Your palms get sweaty, your heart starts racing, etc. Now, of course, there are other parts of emotional expression, and these would include, like the facial expressions that you're seeing here. So there's a whole range of responses that are constitutive of emotional expression, and all of these serve important functions in communicating with others and in preparing behavioral responses when you're engaging in a stimulus that's potentially threatening or rewarding. So the insights from Darwin were also made in terms of using animal models, but also in terms of what an emotional expression involves, were reiterated by William James. And this quote, in some ways, captures certain essences of what emotion is. This is a quote from William James, of course, who was a famous philosopher and psychologist from 100 or so years ago. And he said, "What kind of emotion of fear would be left if the feeling neither of quickened heartbeats, nor of shallow breathing, neither of trembling lips, nor of weakened limbs, neither of loose flesh, nor of visceral stirrings, were present. So all of those things, by the way, are autonomic responses. It's quite impossible for me to think, I say, that for us, emotion dissociated from all bodily feelings is inconceivable. So this is, in some ways, the process. These are the processes that we want to understand about in terms of how the brain mediates these processes. And two very popular theories of emotion were put forth about 100 years ago, or almost 100 years ago, and the debate concerning which of these theories correctly explains how emotions operate in the brain in some ways still goes on. But the notion, briefly, in these two theories is that when you see a sensory stimulus that's potentially scary, like this snake, it's initially perceived. Now, one theory says, okay, you perceive it, and then according to William James and another scientist of that era named Lang, you automatically then express different emotions through automatic pathways somehow, and these would include the visceral or autonomic responses that we just described, and your conscious feelings or subjective feelings of emotion arise from your sensing that indeed you are experiencing emotion, that your heart is beating really fast and you're sweaty. And so this is a theory of emotion that says you perceive it, you respond in a bodily way to it, and then you become consciously aware of it. And the other theory, which was proposed by Walter Cannon and Bard in the 1920s, says you perceive the stimulus first, you become consciously aware of it, and then your brain somehow regulates these emotional responses here. Now, I think the modern day, many people's modern day view is that there are elements of both of these theories of emotion that are right, that there are some brain mechanisms that are very automatic, so that you can trigger certain physiological responses that have to do with emotional behavior really subconsciously, but then there are elements in which the other pathway operates as well. And I don't want to take you through an entire history of the study of emotion, but I will say that nowadays, the way people think about emotion in the brain is they try to understand what particular brain structures are mediating key processes that implement emotional behaviors, and they try to understand how those brain areas do it. And one of the main brain areas that people focus their attention on nowadays for understanding how emotions coordinate in the brain is a brain structure called the amygdala. So this first slide here is showing you a picture of the brain viewed from the side, and this is just a model of the human brain. And if anyone wants to come down later and kind of fool around with this, that's fine. I'll warn you that you can take this whole brain apart when you do it, and it breaks into very little pieces. And anyone that does that is going to get a test during the break. They have to be able to put it back together, or I'll get in trouble with my colleagues at Columbia. But the challenge is, of course, is that this is a big object. Of course, the top is the cerebral cortex, which is responsible, as Eric was telling us, in part for many of the cognitive functions that we do. The amygdala, which is the part of the brain that many people think is responsible for coordinating emotional behavior, is buried in the brain. So if you take this sideways view of the brain here, and then kind of split it down the middle, so you're looking from the middle out, you can see the amygdala located right here, right near the hippocampus, which Eric also talked about. The hippocampus is important for learning and memory. The amygdala is important for learning about emotional things and for coordination of emotional behavior. If you take a so-called coronal slice through the brain like this, you get a picture of the brain here, and you can see here's the amygdala buried all the way deep down in the so-called medial temporal lobe of the brain, one on each side of the brain. Now, the amygdala is a brain structure that has many different subcompartments. These subcompartments are called nuclei, and these different nuclei are specialized to do different things that have to do with the coordination of emotion. There are nuclei that are called the lateral nuclei and the basolateral nucleus and the basal nucleus. This is where sensory information about the world flows into the amygdala. Information about what you're seeing, what you're hearing, about what information about what you're touching comes in and converges in the amygdala, and then the amygdala is poised to be able to coordinate different emotional responses. It integrates that sensory information from many different modalities, and then it can coordinate emotional responses. It can coordinate many emotional responses through output pathways that come from the central nucleus and go down to different brain structures that do things like make your palms more sweaty or make your heart beat faster. And I'll go through some of those pathways as the talk goes on. So the question that is before us, if we have a targeted brain structure that we want to try to understand the function of, and then how are we going to go do that in an animal model of emotion in the laboratory. And so the real way that people have made progress in this kind of question is by using tasks whereby they can manipulate emotional states of animals in the laboratory so that they can study how the brain mediates those different emotional processes. And so the question is, how do we manipulate emotions in the laboratory setting? And in many ways, by far, the most powerful way of doing that has been using essentially conditioning procedures. So reinforcement learning is the process of learning the relationship between stimuli or actions and reward or punishments. And this can be used to regulate emotions. So if you think about this, this is made most classically true with the work of Pavlov, where you just do classical conditioning. You pair a bell, for example, with food reward, and you'll get an animal to approach it because it generates a positive emotional state to the animal. So these classical conditioning procedures can be both appetitive and aversive. In an aversive procedure, you might pair an arbitrary conditioned stimulus, which in this case is a tone, and you'd pair it with electric shock. And when you do that pairing, you would normally get freezing or fleeing from the animal, and I'll go through this in more detail in a few minutes, so that the CS alone, the conditioned stimulus alone, can produce these behavioral responses because you're predicting that a shock is going to be coming in a few minutes. Alternatively, you can do appetitive classical conditioning, where now you pair a tone with a liquid reinforcement, and you can get approach behavior like Pavlov in the dog. And these types of tasks can be very powerful methods for manipulating emotional state in the lab. Now, it turns out, using this type of paradigm, people have shown that the amygdala is very important for learning these associations between stimuli and reinforcement. So this is a classic set of experiments done in patients, and they were using to index learning about the emotional significance of the stimulus. They're using something called a measurement of skin conductance responses. And this is where you just put a couple of skin surface electrodes on a patient's hand, and you measure the current that's flowing between their hand. It's the same thing as a lie detector test. This is what a lie detector test is. It's a measure of how sweaty your palms are, and it's an index of emotional arousal. And so they were using this measurement while they do a conditioning experiment in patients. And in this conditioning experiment, there would be one conditioned stimulus, this blue square, that's paired with a very aversive sound, an air horn that's blowing loudly, and 3 conditioned stimuli that are paired with nothing. So this is the conditioned stimulus that's called a CS+. These are all called CS- because they're not paired with reinforcement. If you if you measure skin conductance response while you're presenting these CSs after the learning, and you do it in normal patients, when you present the air horn alone, you get elevated skin conductance response, right? It's like an emotional response to that very unpleasant stimulus. But when you present CSs here and here, you don't elevate, CS minuses here and here, you don't elevate the skin conductance response because it's not emotionally charged. But then when you present the CS plus right here, you get elevated skin conductance response. Now that's in normal controls, right? So this is not a response to the air horn, it's just a response to the CS plus that's been paired with the air horn, and they're doing that in anticipation of hearing the air horn. Now, if you take a group of patients that have had amygdala damage as a result, for example, of a stroke, and you do the same experiment, what you see is that you no longer are elevating your skin conductance response as when the CS plus comes on. In other words, losing the amygdala, you lose the ability to learn the emotional meaning of that blue square. Okay? So this is evidence from humans that lesions in the amygdala can result in specific deficits in emotional expression that occur as a result of conditioning. And additional evidence that people have found in humans linking the amygdala to emotion, and really there's a lot of evidence now, but I'll just mention a couple of them. One set of evidence that's particularly interesting has to do with the rare opportunities that people get to do experiments in humans who have epilepsy. And a lot of times when patients have epilepsy, their foci of seizures in the brain is down in and around the amygdala. And so neurosurgeons are taking little tiny wires and putting them into the brain to measure the activity in the brain as part of the therapeutic treatment for those patients. And when they do so, they can test the functionality of the brain tissue by applying little stimulating currents to activate neurons at that location. And when they do that in locations where the wire is placed in the amygdala, they can generate feelings of fear and apprehension on the part of in the patients themselves. So that's further evidence that the amygdala is involved in emotional expression then. And then there are some very rare patients that have a disease that very specifically affects the amygdala and the amygdala alone. When you have a stroke, It's very rare actually to affect only the amygdala, because when you have a stroke, you're cutting off blood supply to an entire piece of tissue. It may include the amygdala, but it may also include some nearby brain structures. But there's a rare disease called Urbach-Verhette disease, in which you get calcification specifically in the amygdala and not in the neighboring structures. And if you look closely behaviorally at those patients, they have great difficulty in doing certain tasks, such as discerning the fear expressions in people. So they have deficits in emotional processing, especially related to fear. And this is not a result of the patients having difficulty in visual discriminations. Their visual discrimination capabilities are intact. So these kinds of lines of evidence, including now many neuroimaging studies linking the amygdala to emotional behavior, have led scientists to be quite confident that the amygdala is intimately involved in coordinating different emotional behavior. In parallel to these studies in the human, there's been extensive work in animal models studying emotion and the amygdala's role in it. And by far the most powerful and longest tradition of work has been actually done in rodents using a form of fear conditioning similar to the conditioning that I just told you. And perhaps the most famous investigator using this is actually a colleague of Eric's at NYU named Joe Ledoux. So in fear conditioning, and this is now again just classical conditioning, but I'll give you an example of how people can study it in the rodent. You'd put a rodent in a chamber here, and you play a noise. And while you're playing the noise, you're measuring two things. You're measuring his blood pressure, that's shown on the y-axis, and you're measuring the cessation of movement, or freezing, how much they're moving. And when you play a tone alone, you don't get very much cessation of moving. In other words, they're still moving around in the chamber, and the blood pressure is relatively low. Then you pair the tone with an electric shock that's applied to the grid that the animal's feet are on. So now the tone predicts that the shock is coming, but here we're just measuring the response to the shock itself. And when you measure the response to the shock itself, you see that the blood pressure goes way up. That's another autonomic response, by the way. And the animal freezes, like they're really afraid, right? So they cease moving. That's why that plot goes up. Okay, so the question is, does pairing this tone with the electric shock cause the animal to learn anything? So then they go and do a third experiment here, and what they do is they just present the tone alone again. So this is the exact same stimulus that was presented over here, and the only difference between this experiment and this one is the fact they've had this intervening experience where the tone actually predicts a shock. And when they present the tone here, because the animal has experienced it here, the animal's blood pressure goes up and they freeze. And this is even though they're not even receiving the shock right here. So this is an example of fear learning. The animal has learned that this tone predicts a shock, and they respond in an anticipatory way to the threat stimulus that's coming, the electric shock, by having autonomic reactivity and behavioral manifestations of fear. And using this kind of paradigm, Joe and many other investigators have shown that if you lesion the amygdala, the animals do not fear learn. In other words, if you did this exact same experiment in an animal that did not have the amygdala, you would not see elevated blood pressure and increased freezing here after this conditioning procedure. The amygdala is required for that kind of learning. Now, it turns out that the amygdala, when you're, say, very afraid, is modulating many different things that are constitutive of emotional experience. And this slide is meant to show you some of them. People have done these experiments by now in animals, taking one of these stimulating electrodes that I told you sometimes is implanted in humans, but now implanted in an animal, and you plant it in a certain part of the amygdala, the central nucleus of the amygdala. This is the part of the amygdala that projects to a whole variety of different subcortical pathways, to a whole variety of subcortical structures. These are structures that are down here buried in this brain that I asked you not to take apart if you come and look at this afterwards. But if you stimulate the amygdala in the central nucleus, you'll activate neural pathways going through some of those subcortical structures, and you can generate a whole different range of behavioral and physiological responses that are constitutive of emotional expression. So this work in humans and in rodents makes it very clear that the amygdala is extremely important for many types of aversive emotional experiences. But the question is does the amygdala actually tell us more than that? Is the amygdala involved in telling us how we're feeling? So the data so far indicates that the amygdala is involved in aversive processing and that it's required for learning about threatening stimuli. But is the amygdala just specialized for fear, or does it tell us how you're feeling? Does the amygdala tell us about good as well as bad emotional feelings? And so as we transition here, we're going to transition to some of the work that's been done in my laboratory to explore that sort of question. And in my laboratory, we use a different type of animal model. We use an alert behaving monkey, which of course has many advantages to rodents if we're trying to understand human brain function and human brain dysfunction, because monkeys' brains are much more similar to our own than a rodent's brain, and monkeys' behavior can actually be quite sophisticated and comparable to our own behavior. So when we study things in an animal model that's a monkey, we're much closer to what human brain function is like. So in this experimental preparation that I'm going to tell you about, monkeys are essentially trained to play very simple video games. And so they're sitting here viewing a video monitor. It's not shown on this slide. And while they're sitting there viewing that monitor, we can deliver rewards to the monkey. These would normally be liquid rewards, for example. We can deliver aversive stimuli to the monkey. Now, because we need the monkey's cooperation for these tasks, we use very mild aversive stimuli. These are not aversive stimuli that are like the electric shock that I just talked about for rodents. The aversive stimuli that we would use would be things like air puff. And air puff is a very annoying stimulus to a monkey. He doesn't like it. It's actually very annoying to you, and you wouldn't like it if I put you in there too. And people have actually done experiments in humans where you blast air at them as the unconditioned stimulus during Pavlovian conditioning. And when you do that, you can actually get autonomic responses of this, in response to the CS through that learning. So it's a very effective aversive stimulus on the one hand. On the other hand, it doesn't cause any tissue damage, and you can give it time and time again. So that you can give rewards and punishments, which means we can do conditioning experiments in the monkey. And while we're doing that, we can interrogate what their brain is doing and try to learn about what information is being processed in the brain, how is it being processed, and how might it be telling us about what the monkey is feeling. So in the actual experiments that I'm going to tell you about today, we wanted monkeys to learn the relationship between sensory stimuli and reinforcement. And so we wanted to use conditioned stimuli, like we've told you about before, and unconditioned stimuli, like I just told you about. But for conditioned stimuli, we wanted to use stimuli that were devoid of any meaning before the experiment began. And so we used these computer-generated abstract images. These were new in every single experiment. We specifically didn't want to show the monkey bananas. We didn't want to show the monkey snakes. We wanted to take control of the emotional meaning of these stimuli through these conditioning procedures. So the key question is, if we have a monkey sitting there learning the association between these kinds of abstract images and reinforcement, how do we know what the monkey has learned, right? Well, how would we know that in humans? Well, we have at least 3 ways that we could learn about it in humans. One would be that we just ask the human being, and we can't do that in a monkey, right? So we don't have language available to us. The second is we could measure psychophysiological responses. These are things like the skin conductance response. And you can actually measure these things in a monkey, but it's hard enough to measure individual neuron responses, which is what I'm going to tell you about in a moment in a monkey. So in general, we don't try to do that on top of the individual neuron measurements. Instead, we try to measure the monkey's functional behavior. That is to say that we generally infer things about whether people like something or dislike something by looking at their behavior. Do they approach something, or do they defend against it or try to avoid it? And we know that if they are approaching it, it's rewarding to them, and if they're trying to defend against it or avoid it, it's actually aversive to them. And so we're going to use functional behavior as our assay of what these stimuli mean to the monkey. Here's how the task actually works for a monkey. Initially, the monkey fixates. Then one of these images comes on the screen. It's on the screen very briefly for about 1/3 of a second. It goes off, and there's a temporal gap, which is called the trace interval. So this is a version of classical conditioning called trace conditioning. So there's a temporal gap. And then after the temporal gap, you give reinforcement, either reward or an aversive stimulus. So on the very first trial, the monkey might see that image, and then it gets a reward. On the next trial, it might see a different image, and that one's paired with the aversive air puff. The next trial might be another aversive air puff trial. There's a third image in here, which is actually paired with nothing. The monkey might see that one. And it keeps going until the monkey learns these associations. And 2 behaviors tell us that the monkey has learned the association. One is that the monkey starts to lick in anticipation of reward. It's doing so after this image. And the other is that the monkey starts to blink or close its eyes in anticipation of the air puff that's being blasted at their face. And it does it in response to this image. Now imagine you're recording the activity of individual neurons in the brain while the monkey's doing this task. Now, when neurons are communicating information to other neurons in the brain, they're doing something called firing action potentials. An action potential is an electrical signal that propagates down the length of the neuron, and then it results in chemical release at the end of the neuron, and that chemical actually communicates with the next neuron in a circuit in the brain. So if you're recording how many action potentials a cell is firing, and at this point of the experiment, you're firing more action potentials in response to this stimulus compared to this stimulus, you have an interpretive problem. You actually have an experiment that you cannot interpret. And the reason is the following, is that there are 2 things that are different when you're looking at that difference in neural activity. One thing that is different is that this image is different from this image, and the difference in firing rate that you're observing may simply be due to the different CSs that are on the screen. And the other difference has to do with the different reinforcement contingencies. This image predicts a reward, and this image predicts something aversive. And so you can't tell those two different hypotheses apart just looking at things at that stage of the experiment. You need to do an experimental manipulation to try to understand whether the cell is encoding something about the images or about the reinforcement contingencies. And so what we do in the middle of this experiment, and you heard about this a little bit from Eric, where they're doing something called reversal learning. This is reversal learning all done in the same session in the monkeys, right? This is one of the things that you can take advantage of the monkeys' behavioral flexibility on. So without warning, we'll just flip the meaning of these two stimuli right in the middle of the experiment. And now the image that was associated with something aversive before is associated with something rewarding, and the image that was associated with something rewarding is associated with something aversive, and the monkey learns these reversed contingencies all on the fly in the same experiment. And we can look at changes in neural activity specifically in relation to the changes in reinforcement contingencies to ask whether these neurons are encoding information about the reinforcement contingencies. So what we're doing here is we're snuggling our little microelectrode. This is just like the electrodes that people put in human patients with epilepsy when they're into brain areas. But here we're snuggling it up near individual cells in the brain and listening in on what the cells are doing. We're recording the action potentials that the cells are firing while the monkey's actually doing this task. So one question would be, how does learning about these reinforcements work in the brain? What sort of signals should we look for in the amygdala if it's encoding information about these kind of reinforcement contingencies? And this is a simple diagram depicting another type of conditioning experiment that plays out in my lab all the time. So this is an image of the conditioned stimulus used in my lab. It's me. I'm the professor. Here's a couple of the students in my lab. They're depicting themselves perhaps a little bit in a better way than they actually look. And on Monday, I come in, and the students have a representation of how much I'm worth, how good I am. That's what I'm calling here V of T. It's value as a function of time. They have that representation every day, right? And they also have a representation of how surprised they are by anything I say or do, right? So if I come in on Monday and I all of a sudden say, I've been thinking of a new analysis that I want you to do, well, that is, oh no, not again. So what happens there is my value doesn't change and they're not surprised, right? That's just another routine day in the Salzman lab. I come in on Tuesday and the same thing happens. I've got another new analysis for them. Oh no, not again. Nothing changes yet again. On Wednesday I come in, same deal. They're really bored with this whole drill. But then all of a sudden on Thursday I come in and say, good work team, let's go have dinner at Charlie Trotter's. Okay? I know it's closing, but let's go have dinner there. And so what happens when that happens? They are surprised, right? This is outside the area of routine. So the part of their brain that's representing whether or not they're surprised shoots up. And what is that? That's called an error signal, actually, because it's different than what they were predicting. They had a certain prediction about what they would get when they see me, and all of a sudden now they're super surprised. And the idea is that that error signal is going to feed onto neurons elsewhere in the brain that are encoding how much I'm worth, whether I'm good or bad. And all of a sudden, my value goes way up. And when they encounter me in the future, I'm this really good guy. Okay? So our idea is that the amygdala is doing something like encoding something like value, how good or bad things are. And the question is, so on this other conditioning experiment that we're doing, do neural signals look like this or not, right? And the answer is that they do. And I'm just going to show you a couple of examples of that here. There's two different single-cell examples that I'm showing you, one on the left and one on the right. And what we're plotting here is firing rate, which is how many action potentials those cells are firing as a function of time with respect to when those conditioned stimuli come on the screen. And the first dotted line here is when the image comes on the screen. The second dotted line is when the image goes off the screen. And then the rest of it is that trace interval that I told you that sits in between the image presentation and the actual reinforcement. So we're not showing you the reinforcement responses here right now. So when image 1 was rewarded, this particular cell gave you a big response during this trace interval. And in the initial part of the experiment, we're also showing image 3, and it's paired with something aversive, and the cell fires very little. Then, without warning, we switch the reinforcement contingencies. So we're presenting the same images, but different reinforcement contingencies, and the response of the cell completely switches. So now, all of a sudden, the cell is responding a lot to image 3 and not at all to image 1. So what does that mean? It means that if you're just an ideal observer and you're looking at the activity of this cell, you are seeing that this cell is telling you about how good or bad an image is. It's not telling you about the image identity. It's telling you about how good or bad it is, because it fires more when an image is paired with reward than when the same image is paired with something aversive. Now, we just spent the first part of the talk talking about how the amygdala is important for aversive processing, but it turns out cells like this that respond more when something is rewarding than when it's aversive are present in abundance in the amygdala. So the amygdala appears to be involved in telling you also about how good things are, not just how bad things are. It would be really surprising if we only found cells like this and didn't find cells telling us about how bad things were, but we do find those cells as well, and that's shown here. This is another single-cell example where the cell's now responding more when an image predicts something aversive. That's the red curve here, and it responds less when those same images predict something appetitive. So this is what we call the negative cell. This is what we call a positive cell. And the amygdala is just chock full of these positive and negative cells. It's sitting there telling you about how good or bad things are, just by modulating the activity of these cells, telling you about goodness and badness. So I'm actually going to skip through the next few things because I think I'm running a little low on time, and just move on to the summary of what I told you about. So the amygdala is a key brain structure for regulating our emotional responses. Neural activity in the amygdala can essentially tell you how good or bad things are. These neurons are, if you could just read out the activity of these neurons, they could tell you how you were feeling. And amygdala neurons will rapidly adjust their responses during learning. So even when you change the meaning of a stimulus, the amygdala keeps track of it really, really well. They learn new associations and they keep up to date when the world changes, like when we do the reversal and reinforcement contingencies. And so this makes it a very good structure for coordinating emotional responses in a very faithful and rapid way as you really adapt to the world around you. Now, none of this work would have been possible without a talented group of students and postdoctoral fellows, and the different work I told you about today was actually done by these different students in my lab over the years. So thank you very much for your attention, and I'll be glad to take questions and also to discuss things later. Yes?
You talked about micro-stimulation of the amygdala. Can that be used to help with treatments or?
Yeah, so that's a great question. So, one of the things, so what microstimulation does when you put it into, when you apply stimulating current is it basically will activate all the cells within a certain radius of the tip of your electrode. So one of the things that you require in order for microstimulation to be effective in treatment then would be for neurons that are near the electrode tip to have very similar properties. Now, when people have applied stimulation in the human patients that I told you about, they're applying quite large currents, and in general, they tend to see things having more to do with aversiveness than appetitiveness, and that may have to do with the exact location of the electrodes as well as what neurons happen to be near the electrodes when they're doing those experiments. Deep brain stimulation in general is a technique that's receiving increasing attention for treating certain psychiatric disorders, especially very severe depression. But the location that people go to apply those stimulation is not in the amygdala, where there's this mix of cells that are, some of them specialized for aversive things, some of them specialized for appetitive things. Instead, they tend to go to a part of the prefrontal cortex that seems to be more specialized for making people feel better. And so they actually will implant these electrodes in human beings and drive those neurons somewhat chronically to help alleviate their depression that has basically been non-responsive to all kinds of drug therapies, often non-responsive also to electroconvulsive therapy. It's a treatment of last resort, but it's an amazingly effective treatment in certain examples. Yes?
And could these stimulants be done using some sort of neurological pacemaker in some way?
Yeah, so I mean, that's essentially when they implant an electrode in a human patient, they're just not applying the stimulation once. Essentially, it's like having a little pacemaker that's applying light stimulating current to those brain circuits on a regular basis. That's essentially what they're doing. But the success of that kind of technique really relies on what the organization of the neurons and the functional properties are of the neurons near the electrode tip. So you've got to get it just in the right spot. I don't think the amygdala is going to be the right spot for that, but this part of the prefrontal cortex that people are exploring, it appears to be a stronger possibility. Yes?
I think this may have been in some of the slides that you had to skip over, but could you tell us a little bit about, are you looking at how the amygdala stores those states and all, like how it remembers what's what?
Yeah, so that's a great question. So, one of the limitations of the electrophysiological techniques that my lab uses, so electrophysiology refers to when you're using these microelectrodes to record brain activity. One of the limitations of it is that you generally can you can isolate an individual cell and record from it for a certain period of time, but being able to record from the same neuron over a long period of time is difficult. And so we're not really able to test things, say, over days to weeks to months and be sure that we're recording from the same neuron. So that's a limitation of that technique. That said, in the rodent, for example, you may not be measuring the activity of individual cells, but it's very clear, for example, that the amygdala is also involved in storing fear memories on a longer-term basis. In some ways, that's some of the work that Eric and his colleagues and Joe Ledoux and others have been doing over the last 10 or 20 years. So you can use other techniques to establish that a brain area or a set of neurons is involved in both in the long-term memory, but electrophysiology is a tougher technique to use to establish that. Yes?
Is there anything interesting or unusual about the shape or the structure of the amygdala and some of the neuropsychiatric disorders you talked about?
Yeah, so I mean, the amygdala is a complicated structure. People frequently, and I glossed over this a little bit, frequently talk about it like it's one brain structure, but it's really this collection of nuclei. And so then the question is, what's the differential function of these nuclei, and can some different psychiatric disorders be essentially assigned to the substructures of the amygdala rather than the amygdala as a whole? And I would say that's an example of neurobiology not being there yet. So in terms of understanding the individual function of nuclei, I think in the rodent model there has been some work that is beginning to elucidate the differential functions of those nuclei. I will tell you that I think on a regular basis new work comes out that calls into question the old work in that area, that we still don't have, even the rodent, a good understanding of how the different nuclei operate. So for a long time, people thought the key site of plasticity was definitely the lateral nucleus and not the basal nucleus. There have been some recent experiments that have said actually plasticity in the basal nucleus is probably also important too. So we're still learning about things in the rodent. When you move to the primate, whether it's a monkey or a human, we know very little about the specialization of those nuclei. The one thing we can do very effectively is trace the anatomical pathways of the nuclei. So we know in all these preparations that the central nucleus is the way that the amygdala communicates with these different subcortical structures that mediate different aspects of emotional behavior. That we know from the anatomy. And we know that the basal and accessory basal nuclei project up to the cerebral cortex. And this is, if you think about it, many of our executive functions, whether it's decision-making or perception or attention or memory formation, many of them are influenced by emotional state, right? So the question is, how does the brain mediate that? And so there's an entire area of research that is looking at the influence of the amygdala, not on an emotional expression, which is done through these subcortical pathways, but instead the influence of the amygdala on cortical processing, and the anatomical pathway by which that happens is through the basal and accessory basal nuclei up to the cerebral cortex. There's a lot of projections that come from the amygdala up to the prefrontal cortex. Presumably that's how emotion affects your decision-making, for example. So, I mean, this is all a hot area of research, but it's an area of research that I think is in its infancy.